Okay, good morning everyone. Today's lesson is the third instalment of the lessons we did on the chemistry of food. So this is B3.3 still, but it is the final one. So we looked at carbohydrates and lipids yesterday, and today we're going to look at proteins. So as it is the same sequence of lessons, the learning objectives are the same as the last two. So defining the term macromolecule and giving examples, explaining the properties and uses of the three main macromolecules, and comparing and contrasting the three macromolecules used by the body and explaining why they're an essential part of a balanced diet. There's quite a few keywords today in today's lesson, big topic. The only word I didn't put on the keyword slide is, where is it? Essential. I didn't include that on the keyword slide because you should all know what essential means. Basically, it means you must have it. If you don't have it, then you could potentially become ill or die. It's really crucial that you have a certain type of amino acid, otherwise you will not survive. So that is what that means. You'll probably recognise ribosome from when you did on cells. So we'll go through that in a minute. And the other really important one that you need to know for today is denature. That is a new word. Some of you might not have heard of it before, so really do pay attention to that word. You will need to use it in exam papers as well. So let's just go through the keywords. So we've got amino acid, which is a molecule with at least two functional groups and can bond with other amino acids to form a protein. So an amino acid is a monomer, and a protein, therefore, is a polymer. So remember we looked at them before in the carbohydrate and lipid lessons. So that's a monomer. The protein is the polymer of an amino acid. So the monomer, remember, is the simple form, so it is the building block of a larger molecule. A ribosome is where protein synthesis occurs. That is a really essential process. If you don't have protein synthesis, you can't make proteins, and then you wouldn't be able to survive because proteins are also enzymes as well. So ribosomes are really essential for us to live. Enzymes and hormones I've put under the same sort of bracket. They are both 3D proteins and they have specific shapes for their functions in the body. Hormones are secreted into the bloodstream and are carried to where they are needed by the blood. For example, testosterone, estrogen, and progesterone in the reproductive system. So they are made in the brain and then they are circulated down to the reproductive tract by the blood. So they are taken to where they are needed from where they are made. Then we've got antibodies, which are quite a lot of news stories on antibodies at the minute because of a certain thing called COVID. I'm not sure if you heard of it or not. It's causing a bit of a pandemic. So yeah, there's a lot going around about antibodies and COVID at the minute. So thinking about how to vaccinate people and how to make vaccines. So antibodies are proteins which circulate the body in the blood and they are a part of the immune response. Deficient is the lack of something. So you don't have enough of a specified quantity or ingredient. You often hear vitamin D deficiency. You also have vitamin B2, which can cause a disease called beriberi. That is a really horrible disease, and it can potentially lead to death. So yeah, being deficient is not having enough of something. You can also have vitamin C deficiency, which leads to scurvy. That's when you don't have enough fruit in your diet, which happened to a lot of sailors and people that lived on ships quite a few centuries ago. Optimal temperature is the temperature at which a process is carried out best. Now, this is extremely important for proteins, enzymes, and hormones because they only work at a specific temperature between a couple of degrees Celsius. So if you go above that, they will denature, which is the next keyword. So if you go above the optimal temperature, the protein may denature, and this is where it destroys the chemical characteristics of a biological macromolecule by heat, acidity, or another effect which destroys its molecular conformation. So that means it will not work anymore. So you would have to make a new protein to replace the old one. So proteins, they are used for building the cells and tissues in bodies, including enzymes. Between 15 to 16% of your body mass consists of a protein, or of protein, and there are two types of amino acids. These are the essential ones and the non-essential amino acids. There's about 20 different amino acids in total. Our bodies can make non-essential amino acids, but because we can break down proteins into the amino acids using processes like glycolysis, which we'll learn about if you do AS level biology and at higher levels of biology. But we cannot make essential amino acids ourselves. We must get those from our food. So foods that include a lot of proteins include milk, meat, eggs, lentils and pulses, cheese, and fish. But what happens if you're a vegetarian or a vegan? Because you certainly don't eat this. You don't eat meat, milk, eggs, cheese if you're a vegan, and you may eat fish if you're a vegetarian, but you may not. So the only protein source you've got left is this one. So vegans and vegetarians get their protein from soybeans, lentils, pulses, and fungi. You may have heard of a company called Corn, as in Q-U-O-R-N. But that's not vegan because it contains egg products. But vegetarians can eat this, and it is actually made out of fungi, so modified mushrooms and fungi to make get the protein, as much protein in there as possible. So protein structure then. Proteins are made out of amino acids. So I've depicted amino acids here. These are different amino acids depending on the shape of them. So the squares are the same amino acid, the circles are the same, and the triangles are the same, just to make it easier and more simple for the lesson. And amino acids are made out of carbon, oxygen, hydrogen, but they've also got another element within them, and that is nitrogen. So these are slightly different to carbohydrates and lipids in that they include nitrogen, whereas carbohydrates only include carbon, oxygen, and hydrogen, and lipids are a bit of a strange one. They include a mixture of things, but they're mainly carbon, oxygen, and hydrogen. They don't include any nitrogen. So, okay, so this is how they are in their basic form. You have three separate groups in amino acids. The first group here, I'm going to call group 1. That is an amine group. You don't need to know the names of the groups because that is AS level. I just thought I'd include it. So that's the amine group. That is NH2. So that can be made out of an ammonia molecule because ammonias are NH3. So if you remove a hydrogen from the ammonia, you get NH2, and that can be formed into an amine group, which can bond to the other parts of an amino acid to make an amino acid. The second group here is the R group. So that is specific to each individual amino acid. So each type of amino acid has its own specific R group. You don't need to know anything about that just yet, just that it's there. I don't even think you learn about it at degree level. I don't remember doing that at degree level, learning about the R groups. And then the third group is the carboxylic acid group. So that is group number 3. And that is COOH. And remember, there is a double bond here, which we looked at during the lipids lesson. And that means that there is no need for another hydrogen because all of the oxygen atoms are happy where they are. And you'll learn more about this in chemistry. You might have already learned it already, but it's basically down to the electrons that they have in their outer shells. I won't go into it further now. You can look at videos if you need to. I'll put a video in the link below if you want to have a look at that. But we're not going to go into that in this lesson. Proteins can be made into 4 different types of structure. So you've got, remember there are 4 types. That is important that you remember that. So you have primary, secondary, tertiary and quaternary. This is the basic structure, so that is what these 3 structures are based on. So you must have the primary structure in place, otherwise you cannot have these other 3 structures. So primary proteins are long, simple chains of amino acids, like this one here. I'll just put an arrow there to show you. So remember the same coloured shapes and the same shape of items are the same amino acid. So the order of amino acid determines the function of the protein as well, and this is essential. So this means that there must be a specific sequence to the primary protein structure in order to get a certain protein in the end. So if you had a primary structure that was for amylase, the enzyme that breaks down starch in your mouth, but you wanted to make a hormone for a hormone like insulin, then you will not be able to make that. You will just make amylase because the primary structure is for amylase. So you've got secondary proteins, which are folded into sheet-like structures and have bonding between the layers. These are not really bonds, they're more like interactions, but call them bonding in this instance. So you've got 2 different shapes you can have. You can have an alpha helix or a beta pleated sheet. You do not need to know these names really, not at this level anyway. Okay, so you also have tertiary and quaternary proteins. So tertiary proteins are 3D shapes and they are specific for their function. These include things like enzymes, hormones and antibodies, and quaternary proteins are made up of multiple tertiary proteins which are bonded together, such as hemoglobin, which is found in red blood cells. And a good little fact for you here is there are 4 containing iron, which means that they can carry 4 oxygens per hemoglobin in the red blood cells, which maximises the amount of oxygen that a red blood cell can carry at once. Okay, so proteins and temperature then. As I said earlier, proteins are very sensitive to temperature change, especially higher temperatures. Proteins work best at certain temperatures, and that is called their optimal temperature. This will be around 37 degrees Celsius for proteins found within the human body, because our body temperature ranges from between 37 degrees to around 37.5 to 38 degrees Celsius. So anything above that, the protein will denature, which means that the bonds holding the protein molecules together break and they do not work anymore. So that is when the temperature exceeds the optimal temperature. So that also means that the protein would not work and would have to be replaced by another one. So remember this word because it is very important and it will gain you marks in the exam if you use this word correctly. So denature, remember that. So the functions of proteins then, you've got body structure. Proteins make up muscles and tendons, among other things. Enzymes, hormones such as insulin, and antibodies. But what happens if your diet is protein deficient? Well, there's a disease called kwashiorkor, which is quite hard to say. And that is a disease caused by protein deficient diets. Those most at risk are there's 3 different groups: children and infants in underdeveloped countries, those who live in extreme poverty, and elderly people who live alone and on low protein diets. So that is the theme: low protein diets. Kwashiorkor, if you wanted to look it up, I wouldn't advise it though, it's not a very nice disease. It causes bloating in the stomach, so it looks like a child isn't malnourished, but they are, because essentially what it does is it breaks down your own protein, so you're digesting yourself, which is really sad. And the last thing I want to go over is the food tests. So you may remember looking at these before, but you do need to know them inside out for exam questions. They do like to use practical questions for exams. So watch the video that I will link below. It's quite a short video. It's from another school and they will show you how these tests work. And then try this Google form that I linked here. That will test your knowledge on it. And this is how to, I was going to show you how to get to it. Oh, there we go. So this is how to get to it. You've got the OneNote page. This is the page that I created with this lesson, this series of lessons. You've got the caboodle, which is for the carbohydrate part. So that is your interactive activity. And then you've got the book pages. It is page 40 to 41 for this topic, as I show you here. So that is your pages for today's lesson and yesterday's lessons as well. And you've also got the food test required practical there. And then you've got the PDFs and the PowerPoint. And then here is your assessment that you need to fill out. So if you can either go on here or you can go on the Google form and then you click submit. There is also a Romanian version as well, because I am aware that there is a Romanian speaking student who would benefit from doing her work in Romanian, her native language. So that is available too, which is there. If you have any questions about any of the free lessons or the test or anything at all, oh yeah, there's my Romanian translated PowerPoint as well. If you have any questions about that at all, then please don't hesitate to send me an email and I will get back to you as soon as possible. Next lesson will be on enzymes.